Hi, my name is Andrew Grant and I am a ClinGen biocurator in Heidi Ream's group at the Broad Institute. I'm going to be giving this webinar on behalf of the ClinGen Education Working Group. This presentation will provide a brief introduction into mapping legacy variants to HGVS expressions. In this presentation, I'll go over some of the common issues that curators stumble upon when attempting to identify variants in publications and databases. Oftentimes, when variants are entered into databases such as OMIM, they're entered from papers that are older, possibly from the 90s or the early 2000s, and they will have nomenclature that doesn't mesh well with HGVS. There are also some papers that do not report which transcript the variant should map to, but in order to obtain an HGVS expression, we need to determine which one that should be. Additionally, I'll go over some tips for using the ClinGen Yale Registry and mapping variants to HGVS expressions to get a canonical allele identifier. These tools allow you to identify a variant via the C. or P. nomenclature, as well as interactively generate variants using a transcript. Recently, I've been working on a project attempting to manually map all of the variants in OMIM that are not currently associated with HGVS expressions to the HGVS nomenclature. So far, we've assigned HGVS expressions to over 1,000 ClinVar variants. As you can see in the leftmost column here, these variants are described as one base pair deletions, or P. expressions, or IVS expressions. However, using the information supplied in the OMIM curation, the publication reference in OMIM itself, or even a genome browser, we can identify an HGVS expression and then look it up in the Yale Registry or in ClinVar and see if it corresponds to another ClinVar entry. Or, in this case, if the ClinVar entry does exist for the variant's HGVS expression previously, then we can merge these two entries into a correctly expressed HGVS variant that is only one ClinVar entry. Okay, so here's an example of a variant that has been submitted to ClinVar by OMIM. However, this variant does not have an HGVS expression associated with it. It does, however, have a ClinVar variation ID. So you could use this to start a variant curation or to enter it into the UBR1 gene curation, but this is not a universal identifier. And if you didn't have this ClinVar entry, there would be no way to add these to the gene curation interfaces or the variant curation interfaces. The other issue with this is that the VCI pulls information from computational prediction tools as well as population databases, for example, EXAC and Nomad, but if they don't have an HGVS expression, then it's not mapped to the genomic coordinates, and the VCI cannot pull this information from these sites. So this variant would be a lot more useful to us if it was mapped to an HGVS expression. So in our first example, we'll look at a variant that has been described in a publication as an IVS, which is an intronic variant sequence variant. This old nomenclature was used to describe intronic variant sequence variants when the full genomic sequence was not known. Here we have the SLC26A4 IVS4DS, which means downstream, A to G transition at the +7 site, which just means that it's 7 base pairs from the 3' end of the 4th exon. So if we find a transcript using a genome browser via UCSC or Alamut, etc., and we identify the location that is described, we can then compare that region to any sequencing data from the publication. So if we look at the overview of the transcript in our genome browser, and we see that, and we zoom in on the end of exon 4, downstream end of exon 4, we can try to see if this corresponds to what's being described in this paper here in this figure A that I have screenshotted from this 1999 paper. So starting at what is described in the paper's representation as 639 + 7, if we go backwards to the left, we can see ATTA, ATGG. If we look up above at our genome browser, we can see the same sequence in reverse. So ATTA, ATGG. And we know that this is the exact region that this paper is referring to. So we can take that transcript and take that alteration of the A to T or A to G at that point, and we can write an HGVS expression here on the left, and we can enter it into the Yale Registry. and we can see that this does successfully map to a variant in the allele registry. When we're looking at this, we would also want to see, just to make us a little bit more sure of ourselves, if it does also correspond to a different transcript that may match the numbering on the transcript in the paper. So as you recall, the paper was referring to an amino or a nucleotide somewhere in the 600s. This is at 415. So unfortunately, it looks like this paper was using sort of their own arbitrary transcript to describe the variants in this gene. So we can't be 100% sure that this is what they're talking about, but as long as we have this representation of a variant that matches up the description, we can at least curate the impact of the variant if we want. So next we're going to consider variants that are identified via what I like to call descriptive nomenclature. That is, they've been described well enough to figure out what they are, but don't have full HGVS expressions. In this case, papers and databases will provide just enough information to infer the HGVS expression using a visual tool. So, this variant is the PYCR1 22 base pair deletion variant, and it starts at nucleotide 617 and runs through the intron of a gene. As you can see, the OMIM description calls this variant C.617 to 633 plus 6 deletion variant. So, we want to find a transcript that has an exon that ends in nucleotide 633, so that we can find that exon intron boundary that ends in a C.633. So this first transcript here does not appear to have a exon that ends in C.633. However, if you look at the second transcript, the 6907.3, we can see that the beginning of exon 6 actually starts with C.634, which means the end of exon 5 is C.633. So we know that that is the intron exon site that we're looking for, and this is the transcript that we want. So we will start writing our HGVS expression with this NM006907.3. So if we zoom in on the region, we can find the exact 22 base pair deletion that we want to take out, and we'll actually see that this is an example of the right shift rule. So you can kind of see that there's G's, 4 G's on the left-hand side of the deletion that could sort of be incorporated in deletion, and there's 1 G on the end. And the question is sort of, do we delete the first G, the second G in that amino acid 206 glycine, or do we delete the intronic G at the end? The answer is we want to right shift this deletion and delete the G that is on the rightmost end, so that we will be deleting this sequence highlighted here in red, which gives us the HGVS expression of C.618 to 633 plus 7 deletion. And if the good news is that if you didn't catch up or catch this before you went to go enter in the deletion, the Yale Registry can actually do the right shift rule for you, and it can correct that C.617 to 633 plus 6 into this C.618 to 633 plus 7 deletion. And if you have no idea what the right shift rule is, you can also refer to Stephen Harrison's presentation on HGVS nomenclature that goes over the basics of HGVS nomenclature, and hopefully that will be of more assistance. But for the purposes of this talk, you can just know that the Yale Registry will right shift the variant for you if needed. Okay, so now we're going to get into some of the features of the ClinGen Yale Registry, and I'm going to show you how it can help curators map different variants to the HGVS expressions and canonical allele identifiers. So let's say we have a variant that has been described based on only its C dot consequence. For example, for this variant, we know the gene is PDC10, PDCD10, and then on some transcript, there's a C to T transition at base pair 103, but we don't know which one. So if we go to the allele registry and we click on Gene and Variation-Based Query, it will bring us to this page that allows us to search the registry using what we call the fuzzy query tool. So if we enter our gene, the fact that we're looking at a CDOT, the position of the alteration to the transcript, the reference allele, and the alternate allele, and we click query, it'll bring up all the different variants that have been identified in the allele registry that sort of represent this variant. So this actually will show you that there are 3 different transcripts that are in the allele registry that could conceivably be the variant that you're talking about. But as you can see, the allele identifiers are all the same, meaning this variant is conceptually the same variant across all of these transcripts. So if we select one, we could select any of these, and we go and we click on it, we will be brought to the canonical allele identifier page, where we can get this CA ID up at the top, or we can get the ClinVar variation ID right here highlighted in green, and we can take either one of those and put it into the VCI or GCI. The useful aspect of this tool is if I am doing that project that I mentioned at the beginning and I am looking at this variant and I don't know if it's ever been correctly identified in ClinVar, this will pull up the correct ClinVar variation ID, and then I can submit the request to have this ClinVar variation ID replace the old ClinVar variation ID. Okay, so the other major issue that we often have when we're trying to identify what variant papers are reporting is when they refer to only the protein consequence of the variant. So for example, this variant is a cysteine to serine transition at amino acid position 246 in the LIPH gene. So while we know there is only one transcript in the genome browser that is linked to this gene, we still need to identify the cDNA consequence on this transcript that corresponds with this change. So there's actually 2 ways to do this. The first way, which I'll show you, which is also the one that I think is the most fun, is identifying amino acid position in the genome browser and then utilizing an amino acid codon chart to figure out which base is being altered. So if we identify the amino acid position here, this C246, we see that the sequence is a TGT, and we take that to our amino acid codon chart and we look at the cysteine and we bring it down to the serine, we can see that conceivably a T to A transition at C.736 would convert this or the cysteine, to a serine at this amino acid position. So if we write this HGVS expression, we can have this be a legitimate HGVS expression for this P dot consequence. However, there are some issues with this, and we'll see this in a second when I show you how to do the same exact sort of curation using the allele registry tool. So if we take the same LIPH variant, we go into the allele registry and follow a similar path to when we used the C dot tool, will get us to the same point, but it will be faster and it will be more robust. So if we click this gene and variation-based query and we enter our gene, enter that we're looking at a P dot, enter position 246, enter the reference allele as cysteine, alternate allele as serine, and we query. So what we're going to find is that we actually have 2 different alleles that could conceivably be causing this cysteine to serine change at amino acid 246. So the first that is identified is the one that we had identified previously. We're using the amino acid codon chart to sort of infer the variant change at the cDNA level. And the other is a different way that we could have conceivably identified that same change just based on the fact that the genomic code is redundant, and if we changed a different position in the 3-base pair code for the amino acid, we could have had a G to a C in the middle of the codon that would have made it a serine instead of a cysteine as well. So depending on sort of the level of confidence that is necessary, I'll sort of leave it up to the user to determine whether this is an appropriate choice to make of this first variant, but it is very useful to note that this could be either one of these changes, and it should be important to note that in any curation that you're doing, if you're trying to pull information from the paper that's describing this as just the protein consequence. Next, I just want to highlight there are a few other ways that you can identify alleles and find them in the ClinGen allele registry. So if you go and try to select from the drop-down how you want to query the allele registry, you can see that you can also enter CA IDs, ClinVar variation IDs, ClinVar RCV IDs, DB SNP IDs, exact IDs, nomad IDs, myvariant IDs, another identifier, any one of these identifiers, an HGNC gene symbol, a reference sequence, and a position. And you can also identify a PubMed ID. So this is actually a new feature, the PubMed ID, that leverages the new links that we have between certain variants and PubMed IDs that they have been reported in. So you can search for these variants by the PubMed ID, but it should be noted that many variants have not yet been linked to the PubMed IDs in the ClinGen allele registry, so you may not find any results if you search only with the PubMed ID. For the final example of how to identify or register a variant in the allele registry, I'm going to show you a brief demonstration of how one would go about interactively generating a variant using a transcript. It is particularly useful for indels, or if you would like to skip the step of finding the variant in a genome browser. So if you click on the interactively generate variation link, it will bring you to this page here where you can enter the gene symbol that you are working with. So if you, for example, say USH2A, search the reference sequences, and note that we would choose this long transcript of USH2A, as that is the most relevant transcript based on the biology of what's been studied. We can go into this large text box display, and we will see the entire transcript listed out. And if you highlight a certain position on the transcript, you can, it will auto-populate this field at the bottom for this GAAT. So if you want to delete that sequence, you can leave that alternate code blank. If you want to make an indel, you can enter any reference sequence, any sequence in the position of that GAAT, and then you can search the allele. So if we just take that original deletion and we didn't insert anything, we will get this identifier page, or we come to this get identifier page, and that just means that the allele has not yet been registered in the ClinGen allele registry. So if we want to curate this variant, or we want to add it to a curation, we'll click this get identifier button here, and it will give us this new CAID that we can then bring to any of ClinGen's interfaces. Now, before we finish up, I wanted to point out that some variants cannot be mapped to HGVS due to lack of information provided and/or a multitude of other issues. Unfortunately, for the VCI, if there's no ClinVar ID associated with the variant and you can't map the variant using the ClinGen allele registry, you actually can't curate its impact in the VCI. However, if you are trying to score the variant that you found in a paper during a gene curation and it doesn't map to an associated ClinVar term and it won't map to any HGVS expression in the allele registry, you can actually still add these points at the end of the curation, like so. So if you describe the amount of points that were incorporated into the curation successfully and then you state that the scoring/classification should be changed due to the fact that there is another variant that should be scored. You can note the name of the variant in the explain reasons for change free text box, as shown here. Okay, so to summarize very briefly, if you can identify a transcript and the region that your variant occurs in, you can write an HGVS expression and register it with the Yale Registry. The Yale Registry is useful for quickly providing all the possible transcript impacts of cDNA and pDNA variants, and for complex indels, it is sometimes easier to interactively generate the variant using the Yale Registry. I hope this has been a useful introduction to mapping HGVS of legacy variants. Please visit www.clinicalgenome.org for additional educational resources and learn more about ClinGen initiatives. Our funding sources are listed at the bottom. Please feel free to contact ClinGen by email at clingen@clinicalgenome.org with questions or feedback. Thank you.